Okay guys, we saw in class the nucleic acids, and I'm going to put you this video in the aula virtual to explain the structure of nucleic acid. Nucleic acids are RNA and DNA. The name comes from ribonucleic acid and deoxyribonucleic acid. Instead, they are made of ribose or deoxyribose. You all know that the DNA has a double chain structure, and it's like an helix, and it has some letters inside. You told me in class. Okay, so let's see what that letters are. In the case of RNA, it's not a double chain, it's a single chain, and it has also letters. The letters are 4 different ones in DNA, and they are called nitrogenous bases. Okay? These nitrogenous bases are purines or pyrimidines. Purines are adenine and guanine, okay? And pyrimidines are thymine and cytosine. In the case of RNA, instead of having thymine, okay, in the case of RNA, instead of having thymine, it has uracil. So as you can see, adenine, uracil, guanine, cytosine, and DNA is adenine, thymine, guanine, and cytosine. DNA has got thymine, and RNA has got uracil. Nitrogenous bases are a ring, like rings of carbons, okay, with this structure, and they have a nitrogen atom, nitrogen with hydrogen, as you can see here. Okay, this about the nitrogenous bases. Nitrogenous bases are like the alphabet that contains the information and the genes. Then we have these sticks. You told me in class that these were sticks. Okay, so those sticks are deoxyribose in the case of DNA or ribose in the case of RNA. This one is the ribose, okay? It's a monosaccharide made of 5 atoms of carbons. This is the first one, second one, third one, the fourth one, and here is the 5th one. This is the ribose, okay? This is the ribose, and this one is the deoxyribose. Why do we call them? We call it deoxyribose because this is the ribose and this is the deoxy. As you can see, there's an oxygen left, so that's the reason why we call it deoxy, because there's an oxygen left. This is deoxyribose for DNA and ribose for RNA, and they are the sticks, okay? And then we have the chain or the bone, the backbone or chain. This backbone, these parts are made of phosphoric acids. The phosphoric acid, this has this molecular structure. It's a phosphorus with 3 groups of OH, 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 and an oxygen linked by a double bond. So, I hope so far so good till here, okay? This is the structure, and now we are going to see how do they... Oh, sorry, I forgot to tell you something. The bases are complementary. This means that when in a chain we have adenine, in the other chain we are going to have thymine. When in a chain we have thymine, we are going to have adenine. In this one we have guanine, so here it's going to be cytosine, and where's in the place where is cytosine, here in the other chain we are going to have guanine. Okay, we say that they are complementary chains, and here you have the adenine always is complementary to thymine in DNA or uracil in RNA, and guanine is complementary to cytosine. Okay. So now, here I have the structure, the adenine, thymine, guanine, okay? You can see here, this is the adenine, this is the thymine, and this is the guanine. Nitrogenous bases, which has a stick, which is the deoxyribose in case of DNA, okay? And the backbone, which is the phosphoric acid. So I have the backbone here made of phosphoric acids, okay? And the sticks that joins the phosphoric acid to the base. So how do they bond? Obviously, as you know, when we make a bond, in general, we take out water, H2O. So here we are going to take H2O. So I take this H, this another one, and this oxygen, and it's going to be bonded like this. Okay? And in this case, in the third carbon, this is the first one, the second, and the third, is going to be released water. So we take this hydrogen, these hydrogens, H2, and the oxygen, H2O. We release water, and it's going to be bonded. This oxygen is here, like this. Okay? So, if we do the same with the other ones, we are going to have this, the base, joining to the deoxyribose joining to the phosphorus, and in the last one is just the same. Okay? These are called nucleotides. This is a nucleotide, this is another nucleotide, and this is another one. And now we have to join this first one with the following. So, this phosphoric acid is going to join with this deoxyribose by the fifth carbon. Okay? This is the fifth carbon. So, it's going to be CH2OH. So, we take out this, and we take out this, and we take water, okay? And it's going to be joining like this. And the same with the other one. In the fifth carbon, take this, the oxygen, and it's going to be like this. Okay? And now, I have told you that they join by the third carbon and by the fifth carbon. Okay? This is, take a look on this. It is this one is joined with the fifth, and to the other, and to the next one by the third one. So we are going to have a polarity in the chain. So the chain here is joined by the fifth carbon. This one, the fifth, is free for joining with another one. So we say that this is the fifth extreme. Okay? And in the other one, this carbon is the third, and is the one that can join to another one. So this extreme is going to be the third. Okay? We can make another chain, okay? The parallel chain. Here, where is guanine, here it should be cytosine. Thymine should have adenine, and adenine should have thymine, okay? And the same, they would be joined with the deoxyribose and the phosphoric acid. And it would have, this comes from the fifth to the third, okay? So in this case, it's going to be from the fifth to the third. And so this is all, and I hope you haven't got any doubt.